Welcome to Human Fetal Lung Cell Atlas. This tutorial is going to go over the basic functions of the Human Fetal Lung Cell Atlas web portal based on cell by gene. Here we are sharing for types of data, single-cell RNA, single-cell ATAC, Visium and in situ staining. All of these data are produced by the work listed here. We will start the tutorial using the single-cell RNA-seq platform. As an example,
first click the View Single-Cell RNA-seq button. And then you will see multiple modules on this page. You can click the button to view the interactive viewer, or to download the H5AD files corresponding to each of these modules. We have an overall module for all of the cells, and also the individual modules corresponding to different lineage compartments or subsets. First, let's look at Interactive Viewer. If it's your first time to open this browser, it might take a while. On the left panel, you can see the metadata that are listed. These are the categorical metadata, which means every cell is put into a discrete bin or category. For example, if you want to check the batch, click the water drop, and then the colors will represent the batch information. By clicking the arrows, you can see the breakdowns and the corresponding colors of each of these categories. Similarly, we can also let this Self-Steam browser show the big clusters and more fine-grained broad types, and the version of kits we use to generate the library. The dissected part, the donor pseudo-IDs and inferred sex, and immunoreceptor isotype summaries. Leiden clustering, and the most fine-grained cell types or states. If we move the cursor above the labels, then the corresponding cells belonging to that label will be enlarged. And if we want to make the other cell types smaller, we can deselect all of the cell types and then select the type of cells you are mostly interested in. For example, the erythrocytes here. And to remove all of the other cells from the view, we can click the subset button. So now you will be only seeing the erythrocytes that you are interested in, and click the full pie to restore the setting. If you want to show the labels on top of this embedding, you can click the label button. So now you can see all the labels on top of the cells.
If you want to see the contribution of different batches to this ArchR cluster, then you can click the water drop of the batch, and then move back to this erythrocyte. Now you can see the proportions of the different batches within this erythrocyte cluster. And by clicking the subset button, you will be only keeping the erythrocytes. And then if you go back to the batch, you'll see the numbers breakdown for each library.
Now let's go back. If you continue to scroll down, you will see more categories, as well as the continuous variables. Here are the histograms of each continuous quantitative metadata. If you click the water drop, you can see the heat map representing the quantitative level of one variable at a time, such as the developmental stage, percentage of mitochondria reads, the number of UMAP counts detected, the number of genes detected per cell, and other scores. We can also clip the continuous metadata, which is to limit the range of the metadata, such as the percentage of mitochondrial reads. We are showing the bottom 20% of the cells in terms of mitochondria fraction, and we can revert it back to 100%.
And on the right, we can check the expression of individual genes. For example, we type EPCAM, which is a gene for epithelial cells. And as you can see, it is enriched in the two compartments of epithelial cells. We can show the histogram of the gene expression in individual categories, such as the batch and the broad cell types. At the bottom, we can see the current embedding. By clicking on the embedding, we can switch between different embedding coordinates. For example, currently, we are showing a collection of 9 separate UMAPs, juxtaposed side by side. But we can also show a single big UMAP, containing all of the compartments, by switching to UMAP Original, and click again to hide the menu. Now you see the overall big UMAP, and we can again show the same type of categories, using the left buttons.
Now let's come to the right panel. If we click the expand, then we will be able to select the range of expression that we're interested in. Here I'm interested in the cells that are expressing higher levels of EpCAM. Now the cells with EpCAM expression in this range will be highlighted or enlarged. Here, again, if I click the subset, then only the EpCAM high cells are kept. Click to revert back. If I'm interested in the list of genes, I can create a gene set. For example, I will create a rudimentary gene set of epithelial markers. And then I will add the marker genes one by one.
EpCAM, keratin 5 and keratin 14.
And then I click Create Gene Set. Now if I click the water drop, it will show the summary score of the genes in this gene set.
Additionally, you can also make a scatter plot with x-axis as one gene and y-axis as another gene. Now here you see a scatter plot. One dot is one single cell. You can click to hide and show the scatter plot or remove it. If you are no longer interested in any of the genes here, you can click the bins to delete them. You can also delete the gene set here.
Finally, there is a very important function, which is differential gene expression. While Cell by Gene was created as a visualization tool, it can also help us to perform quick differential analysis between two populations. And this can be done in two ways mainly. One way is to use this selection tool to circle out the part of cells you are interested in. If you are interested in this island of cells, you can select them and mark them as population 1. It has 13 cells in it, and then you want to see the difference between this population and its neighbors. You will highlight this neighbor zone and register as population 2. And then you can click the calculation. Now you get to lists of genes, the POP1 high and POP2 high genes. By expanding it, you can check out individual gene expressions. Another way to select the two populations is by selecting the right metadata. Easy.
Now let's go back to the front page. Enter single-cell RNA. In addition to these filtered fetal lung and organoid single-cell data, we also provide the readers additional data sets, including the not-filtered data, so that you can inspect the artifacts, in case you think there are interesting populations filtered out. Additionally, we have also included other companion datasets, such as the fetal heart dataset, to compare the 5' and 3' protocols we used. We also included the 3' fetal lung single-cell RNA data here.
Now let's go back to the home page. In addition to the single-cell RNA-seq data, we also have included the single-cell ATAC-seq. Visium data, and also single-cell in situ staining images.
And if you go to single-cell ATAC-seq datasets, you can see a similar view. But this time, we can not only see individual genes' locus scores, we can also see the motif scores of transcription factors and the imputated RNA level based on RNA attack integration, all labeled with a suffix after the gene name.
We also included Visium datasets. If we click this Visium viewer, you will be shown a couple of Visium samples here. You can download them by clicking the download button or view the data by clicking on the images. Now the image is loading. As you can see, it looks like the single-cell RNA interface. But one difference is that one dot is one voxel. And by clicking the image button here, you will be able to see the H and E image. And using the magnifying glass, you are able to zoom in. If you want to hide these dots, you can and select one of these. Now the dots are smaller. You can also do other filtering or clipping tricks to hide all of the dots to see better the H and E image. Finally, all of the in situ staining images are listed here in this table. And you can find the developmental stage and where in our paper it is used, and which genes, transcripts, or proteins are stained here.
If you want to look at a 3D view of the cell types here, this is the 3D UMAP where you can click and rotate. You can also select and deselect individual cell types here to find where they are. There are also a lot of other tools here and you are welcome to explore them. For more information about this paper, please check out our GitHub folder.
This paper is one of our Lung Cell Atlas series.
And if you want to see more information, please go to lungcellatlas.org. Here you can see all of our lung atlasing efforts, including our embryonic and fetal work, as well as other interesting data sets for you to explore. Thank you for watching.
Lung Cell Atlas, we mapped a new multi-omic facial for a breakthrough. With cell by gene, we see secrets profound. Discoveries await where knowledge is unbound.